healthy animals as high quality food resources. I'm Dr. Tertia Needham. I'm from the Czech University of Life Sciences for the online listeners, of course, sorry. I will just repeat, I'm Dr. Tertia Needham. I'm from the Department of Animal Sciences and Food Processing at Czech University of Life Sciences. And yes, so I would like to just remind the speakers that they have 10 minutes for presentations. I will just raise my hand if that 10 minutes is up, and you can wrap it up quickly. So it can leave us with another 5 minutes for questions and discussion. All right, so, and also welcome to anybody who's joining us online. Please don't be shy, send us questions as well. Our technical advisors will help us with that to moderate it. So we have 4 presentations that will be in person over here, and then one online as well, which we will move to the last because we have to switch between the technical equipment for that. So without further delay, I would like to call up the first speaker. I will not announce your title, so you can do so yourself. Yes, so we're going to be looking first at some dairy production. And there's a microphone for you.
Okay. Yeah, good afternoon. Yeah, my name's Asan Dambi, and I am presenting results from research we did together with the University of Nairobi. I work with Wageningen University and Research, and this was part of a project called Africa Milk. Where do I push? There's a present. It's okay. It's not connected. Because no, still not. No. There should be a button. Okay, so it's on the hygiene practices and also the safety of smallholder milk from smallholder farmers in Kenya. And I'll start by presenting the context. Milk is such a big contributor to livelihoods in Kenya. It employs quite a huge number of people in the whole dairy sector. And it is dominated by smallholders who produce about 85% of the total milk. So these are farmers with fewer than 3 cows or 3 and below cows. And there is also the issue of quality because, you know, when you aggregate milk from so many farmers, quality tends to be a challenge. The sector depends on this. And the really big issue is also that there is a huge competition for milk between the informal and the formal sector. So Kenya is not self-sufficient in milk production. It used to be, but now there's a lot of import of milk from Uganda, and there is a lot of competition. So there are processors, the formal sector, and there are also milk vendors or neighbors that buy milk from farmers. And why I am explaining this is because it has an implication, thank you, on the quality of the milk. Because if there is that competition, then there's a tendency that you will compromise on the quality because then you know that your milk will always be bought. And this has happened in many cases where farmers do not really have to make the effort to produce good quality milk, not only farmers, but all the players along the chain. Because if your milk is rejected in one part, you can still sell it if it's rejected by one processor or by one buyer, you still have the chance to sell it to another buyer. And that is what has led to quality issues. And there have been so many concerns raised on milk quality issues in Kenya. So the study we did was just to assess the hygiene conditions of farmers both looking at the knowledge part, what they know and their practices. And also the second was looking at how the milk looks like in terms of quality. So laboratory analysis were done. And there were 3 sites. So there were the Highlands, Bomet County. You can see on the map also the sites. There was Nyeri, Nakuru, and Bomet counties. And as I mentioned before, there were 2 things. First, a survey to look at the practices and if the farmers' perceptions on hygiene. And the second was looking more at the quality of the milk as analyzed based on laboratory. And these were mostly microbial analysis. And I would also emphasize here that we did a lot more, but this presentation is focusing only on the microbial quality. So milk was sampled from farmers directly, but at the milk collection point, and it was also sampled from traders. There were a number of traders who collected milk from many farmers, and the milk was sampled also as some kind of a combination of milk from a few farmers. Milk was also sampled at cooperatives at different levels, aggregated by farmers, also bulked. There are different bulks, bulking before cooling and after cooling. And the milk was also followed right to the gate of the processors. There was also an objective to see the point of contamination if there was any contamination in the milk. So just regarding the practices of farmers and perceptions, we can see that in the first, the use of plastic containers to handle milk was a rampant practice. Well, not as rampant as many others, but still it's not tolerated by the regulations to use plastic containers because of they have the ability to carry a lot of microbes. It was still possible, and we also have no refrigeration of milk before it reaches the collection point. Almost all the milk was never refrigerated before it reached the collection point. And farmers had cows that suffered from mastitis, and they noticed it, but the milk was still delivered. And we also have the farmers that did not separate the mastitis cows. They noticed that the cows were mastitic. They still collected the milk from those. And there were many farmers who did not clean the sheds of their cows. So there was also a possibility to have contamination. And we also saw that there were a number of farmers who had a longer distance to deliver their milk. Most often we measure distance in walking time, not really kilometers. So there were some farmers that had quite a long distance, and we also had some farmers who were using the reusable other cloth, which is recommended, but also not every farmer was using it. And well, this is separated into the different regions, and I think we can see the differences. And just one highlight about the different counties. Bomet is a place where there are more pastoral, more range, so grazing farms, and Nyeri is the complete opposite. They are more zero grazing, so the animals are housed most of the time or almost all the time. And then Nakuru is where there's a bit of a mixture, where there's some grazing, mainly parts of the day or parts of the year, and then the rest of the time they're kept indoors. So regarding the bacterial quality, we had here 4 different tests. We had the total viable counts, E. coli, Listeria, and also Staphylococcus. And what I would just highlight here is that in all the 3 places, you can see the standards here. These are the standards for the Kenya Bureau of Standards. All the values that we had on average were higher than the values recommended. Almost all, I think we just had one case where the value was less than the standard, but all others, the bacterial load exceeded the recommended standard by the Kenya Bureau of Standard. And there were various reasons for that, which I also explained already before. There were poor hygienic practices. We also had hand milking, which is almost done in all farms. And we also had the fact that farmers were not cleaning the sheds and also not disposing the dung regularly. And sometimes animals were allowed to graze and also consume poor quality feed and also poorly preserved silage. Just a recap of the challenges that we noticed on the farm. There was sometimes very poor feed, poorly preserved feed, both concentrates and roughages, poor cleaning of the cow sheds. And then we also had sometimes excessive dosage of antibiotics with the residues in the milk. And then we also had hygienic practice, poor hygienic practices at the farm. And we also discovered that, well, in the bigger part of the study, that there was addition of hydrogen peroxide into milk, which was not recommended, which is forbidden by Kenyan regulation. And even at the level of bulking and processing, from our results, we anticipated that there were still some malpractices that went right to the level of bulking and processing. And as a conclusion, we think that the farmers in all the regions were not fully aware of hygienic practices and did not employ the practices. And this resulted in poor hygienic quality, and they exceeded the recommended standards by the Kenyan rule. And as recommendations, we think that we should have continuous training of farmers, improvement of infrastructure, transportation, and the cold chain for the milk transportation. And we also think that if there is a quality-based milk payment system, which is enforced, then it will push farmers to comply to quality standards, or at least to improve on the quality standards. And finally, regarding the policy environment, we think that we need 2 kinds of policies. Usually people talk of the carrot and the hammer. In some cases, we think that we should do supportive policies that encourage farmers to produce, maybe give them incentives. But we also think that in some cases we should be more punitive, because in some cases farmers, for example, know that they have just treated their cows with antibiotics and there are residues in the milk, but they sell because they say, we have to make an income. We can't throw the milk away. So we think that both sides of regulations are required. Thank you. And that's it.
So thank you very much. So now I'll take the opportunity to welcome questions from the floor. First.
Thank you. That was really, really interesting. One thing that we wondered was that you said at the beginning, one thing you asked for is that if they use reusable cloths and that this was a thing that was encouraged, and we wondered why, because actually reusable cloths, they carry the bacteria from one udder to the next, and then so yeah.
Yeah, usually there are two things. Reusable cloths can be used, but we do not encourage them to use the same cloth for different animals. You use it for each cloth per animal, then you can wash and use a next time. So that's what is recommended, because if you want to use disposable for smallholders, it becomes a lot more expensive.
And rubbish problem.
Some other question that I had is if raw milk consumption in Kenya is used to, or it's usually processed and pasteurized, because in this case also it would be interesting to know what the sanitary status of the animals are, if they're proved for brucellosis or tuberculosis or other agents that could be zoonotic for people.
Yeah. Just on the case of zoonotic diseases, they are rare. They sometimes occur, and it's usually in the news when they occur. So it's something that is detected and like put very fast in the media. And about treatment of milk, it is very common for milk to be heated, to be used in tea, boiled and consumed. There are few parts of Kenya where people still, or some traditions where people still consume raw milk, but I think it's a smaller part of the country.
Thanks for the presentation. So how long it requires to process the milk in the current situation?
Well, processing the milk, it's mostly yogurts, or there's a pasteurized milk and the UHT. Cheese is also done, but it's not very common. I would say how long it takes to process the milk. Usually the milk of the same day is processed. So it depends on the time it gets to the processing company, but it can't stay longer than a day in the processing unit. Yeah, because the bacterial content is usually so high that they can't afford to keep it longer before processing. At least there is a pasteurization that is done on the same day when it is delivered.
Thank you.
I think it's good.
All right, so moving from Kenya to Tanzania, I would like to bring up our second speaker, Saman. Okay, so the floor is yours. And please don't forget to use the microphone. It's somewhere. Yeah, there we go. And keep it up to you.
Okay. Welcome to my presentation. It's about a vaccination campaign in Tanzania, and it's about the adoption and characteristics of East Coast fever vaccination. And East Coast fever is like a really critical disease in Tanzania, and the economic loss is really high. And there is a vaccine available called ITM, which is infection and treatment method. And it gives the animal lifelong immunity once administered. But the challenge is, or one of the challenges is, it needs liquid nitrogen for cooling. And as you may know, in Africa, the infrastructure for like cooling systems is a challenge. And also the price of 10 to 12 US dollars might be a challenge for farmers to adopt to the vaccine. And that's why we have a low adoption. Then what was our question? Our question was, which factors influence the farmers to adopt to this vaccine and to increase the adoption of it? And the second question we come up with is about the differences of travel distances from both perspectives. I mean, the healthcare provider must come to the farmer, and it should be economically feasible that he can come there. So if the travel distance is too long, it may be, well, also an obstacle, but we come to this later. So what does the literature say? We found out that the literature already said that access to credit is important, the travel distance plays a role, and market access and farm income are also important. So now where did we collect the data? We collected the data in Tanzania, and it is the lake region in the north, in the southern highlands, and in, yes, Arusha region. And we have two questionnaires from farmers' perspective and from the vaccinator perspective. And on the map, the blue dots, they are the farmers, and the white dots, they are the healthcare providers. But don't be confused that we have some points like in the sea or in the lake. That's due to privacy reasons, because we did not use the exact coordinates. Okay, what did we do? We separated animal healthcare providers into 3 groups, like in the government group, government and private, like a mixed group, government operators who also have a private business, and a private business only. And then we looked, okay, who is offering ITM? And we said, well, yes, that's the whale and the syringe picture, and no. And then on the other side, here, we looked at the farmer's perspective, which is the farmer using the ITM or not? And what we want to find out is something in between here. So what did we do exactly, or how did we do it? The methods, we choose like the variables from the literature, which you just saw, and we backed them. Then we used descriptive statistics, which I show you in a minute. And then from this, we built 3 statistical models. One is the logit model. The second is the OLS, or ordinary least square model. And at the end, we have like a combination of logit. We have a like censored model, which means we left censored it so that we have cut. We start at 0 so that we don't get negative values. Yes. And now we come to the results. This is the descriptive part, but soon the econometric model is like most interesting. So what do we have? We have the land cultivated per hectare, or in hectare, the amount of land cultivated. And you can see the difference, like in ITM, we had a sample size of 360, and in the no ITM group, 540. So here you can see that in the yes group, we the average cultivated land was 6 hectares, and in the non-user group, 9 hectares. off-farm income is roughly equal or in balance, and the livestock produce value is in the ITM user group higher. And then this large difference between ITM yes and ITM no, well, that's a little question. It may be that farmers produce different crops, and different crops may have different yields. And for our regression models, we took these, or one of also these, but more of these items. And we also divided it into yes and no groups. Yeah. Now we come to the education level. Why is education important when it comes to ITM? You can see that the higher the school, the education level is, the higher is the difference between ITM no and ITM yes. And most important is like the primary level. And why is this education important? It may be that farmers can read and understand like the benefits of the vaccination better, or have access to certain media, like maybe written text and some things like that. Sorry, and then you can of course see a trend that the higher the education, the more uptake, difference you have. And now we come to the travel distances. And as you can see, it stands out that the government-only group in the ITM non-user group is the highest. And why is this? This may be because the longer the distance, the more effort you have for cooling purposes. So you have to meet the cold chain. So when it gets warmer, like the vaccine vials, then they can be done off. Genau. And in this slide, we looked at the aspect of access to credit, which gets in the next slide even more interesting, is that credit in the yes group is like that farmers have like more access to credit in the ITM user group. So these are farmers who already use the vaccine. And in the ITM non-user group, we have a difference between credit and no credit. So in the non-user group, more farmers have no access to credit. And now comes the interesting part. This is our regression model. And we did like the 3 regression models. One is like the adoption practice, which is like for I use it or I don't use it. Then we have the ordinary least square model, and the Tobin model, and this is like the degree or the extent for adoption of this vaccine. And as you can see, the black squares, they are the significant values. And in these, and what really stands out, what is really significant is the access to credit, or like a pay later option. That maybe, okay, I get the vaccine, I can pay maybe a week or 2 or 3 later. And it stands out that it is significant in all 3 models. And then for adoption, we have the distance is important. So OLS means it includes users already. So OLS part is like farmers who already use the vaccine. And to get farmers or to motivate them to take the vaccine, like the distance of the animal healthcare provider is significant. And the value of the livestock produced per cattle count is important for taking the vaccine. And we have the education level, which is important in the TOBIT and OLS model. So if a farmer already uses this, the level of adoption, that means these two are like also factors to consider. Okay. So at the end, what should you take home? You should take home that the access to credit is important as a key driver for the uptake and adoption of the vaccine. And then the travel distance is like a key challenge for the cold chain. So if you have a thermostable vaccine, then you can store it longer and you do not need this high effort. And at the end, we have a share of marketed milk, and farmer education are also important for the level or grade of adoption. Yeah. And that's it. Thank you for your attention, and we are now open for questions.
Yes, thank you very much. All right, so we have about 2.5 minutes for questions. So I will open it to the floor first. Okay, so perhaps I have a couple of questions. Is this part of an ongoing study that you're busy with, or is it completed?
It's actually my master thesis, or
Yeah.
part of my master thesis, and the data is like finished, and it needs to be written still, but it's basically it.
And from what you experienced, do you think there is some opportunity for some like governmental support vaccination program in this case?
Also, such programs exist, but the question is how big are these programs, and do you reach every farmer who is like suffering from the disease?
Yeah, of course, if the travel distance is a huge
Exactly,
factor,
yeah.
of course. Yes, there's a question on the floor.
My question is super easy. Thank you so much for your presentation. I'm really interested to learn a little bit more about like this last mile distribution. What exactly is this distribution method?
So the last mile distribution is like distribution between the farmer and the healthcare provider. So the healthcare provider needs to drive to the farmer with a vehicle and to organize everything in between, like cold chain. This is like the last mile, but also like not a physical last mile, but the motivation to say, okay, I go to the healthcare provider and I want to order, I don't know, vaccine for 100 cattle, probably. And that's like the motivation behind it. Why should I go to the farm, to the healthcare provider and to buy this vaccine? The question is why and what is my motivation?
Okay, thank you.
All right, so we're ready for our third presentation, which is from Glindis Luciano. So please welcome on stage. And now technically we're moving more Southern Africa, but also somehow connected to the Czech Republic. So Glindis, I will leave it up to you. Just want to check if it's the right one.
Hello, everyone. Good afternoon. My name is Glindy's Virginia Luciano, and I'm really excited to present my research on the effect of dry and wet aging on the physical and sensory quality of common eland meat. I was supervised, this is part of my thesis, of my master's thesis. I was supervised by Dr. Tertia Needham, and also I had the consultation of Dr. Dania Budish from the Institute of Animal Sciences in Prague. So we know meat is important. We all kind of eat it. And we also know that the demand of meat is increasing depending on the region. We also know that as a household incomes increases, so does the demand of animal-based products. The meat of game species can be seen as a potential alternative resource that can actually supplement this demand. However, when compared to the meat of domesticated species, we know that the nutritional properties, sensory quality, and other aspects such as safety are not consistent for the meat of game species. We also know that these nutritional sensorial qualities are also very important to the modern consumer. We look at a meat when we're going to buy it. We care about how it tastes. We care how it feels when we're chewing. So these aspects are very important. So a starting point, the common eland has actually been identified as a suitable species for meat production since the 70s. However, upon initial research, has been found that the meat of eland is tough. Although wet aging improved tenderness, the meat was still considered tough, let's say for the average consumer. And so this is where this research comes in to try to look into what can be done if aging, different forms of aging can actually help to improve these qualities. So what are the issues? As I already mentioned, the knowledge that we have for domesticated species really differs from what we know of meat, the game meat, of its processing and its quality. For example, the controls for antemortem factors and postmortem factors have actually not been established. At the moment, carcass yield is actually the most important economic performance parameter, which is an issue for commercial purposes. And for this reason, more research is also needed in order to be able to improve quality aspects. And we cannot just simply assume that what has worked for domesticated species is also going to be able to work for game species. So the research questions are, does aging in general affect the overall quality of farmed eland meat? And how will dry aging compared to wet aging affect the physical, chemical, and sensory qualities? So for the methodology, we had 6 subadult female elands. The LTL muscle was removed and then randomly assigned either dry age treatment or to a wet age treatment for 14 days. Measurements were taken on day 1 and on day 15. So on day 1 and on day 15, we took measurements of the pH, the color, the shear force, which means how tough bite might seem of the meat, cooking loss, chemical composition, as well as lipid oxidation. We also, for the dried aged meat, look at the crust formation and the penetration. We also had a sensory analysis by a trained panel, which I will speak a bit more about. The sensory analysis was conducted by 10 trained panelists, which assessed 15 different attributes. The tasting sets were provided either a wet age sample and a dry age sample from the same animal, but these were randomized. So of course they didn't know which animal they were eating the sample from, and in total it was 12 samples. Going back to the methodology here, then all of this data, of course, was processed using statistical analysis through SAS. So the 10 panelists were really lucky on this day to be able to take a look at the beef aroma intensity, the game aroma intensity, tenderness, juiciness, fineness, chewability, beef flavor, intensity, gain flavor intensity, abnormal flavor intensity, liver flavor, sour flavor. You get they're over here so you guys can see them. And then you can also see how exactly were they evaluated, and then on which on the scale as well. So the results, part one. So as I mentioned, economic performance for game meat is very important, particularly because you're looking at the carcass yield, right? This is how producers are able to make money off the meat, right? Of course, when you have meat such as dry age, unfortunately, you are prone to lose a portion of the meat, right? Through the aging process. And you can actually see in the circle that I was looking at the penetration of all the different samples and did a small descriptive analysis of how much of these trimmings that you would need to cut off, right, before selling it to the final consumer would be lost. This case, on average, across those samples, it was about 16%. That's quite a high number, especially considering that this is already dried age. So the original carcass yield muscle is already decreased, right? And then on top of that, you have to get rid of all these trimmings of these crust formation. However, something that can be seen as an opportunity for producers is that these trimmings can actually be used for other products as well. They can be used to add more flavor, and they could also be used to create other high value products for consumers. So instead of seeing it as a loss, it can be considered as an opportunity. Now on the physical meat quality, characteristics, and chemical composition. So of course, through aging, we can actually see that the pH did increase, which is normal. This is part of the aging process and the breakdown of amino acids. With lightness decreased, however, the redness and the yellowness increased with aging, and values were completely normal for what's standard for game meat species. Of course, you're going to have a higher weep loss, as I mentioned, with dry aging, and which leads us straight into also moisture content, which of course is going to be lower as well for dry aging. And you're going to see also like a higher concentration of things such as accrued proteins, muscular fat when looking at dry aging as well. So the sensory analysis. So remember this very long chart with the 15 attributes. At the end, what we actually saw was that abnormal aroma intensity and liver flavor were scored higher for the wet aged meat compared to the dry aged meat. So this is something that can be seen as a bonus for dry aged meat, particularly liver flavor. Across multiple studies, it's really hard to jot down exactly what this means, but it's usually attributed to gamey flavor, which is not considered a positive attribute, but it's mostly due to the fact that consumers are not trained or accustomed to eating these types of meat in general. And of course, abnormal aroma intensity, as I mentioned, scored higher for wet. And this could also be because when you have dried aged meat, there's like different, there's a unique development of flavors that you have, development even of aromas. So of course, this can be the general reason as to why. Now, so for conclusions and general recommendations. Dry aging indeed showed better flavor and aroma development, especially compared to wet aging, and overall acceptance was not affected. However, with further research, it can be that these factors can be affected. Aging did not affect in any negative way the physical chemical qualities of the eland meat, and a recommendation is that recommendation would be that perhaps combination of different aging techniques as well as alternative technologies should be investigated. An idea would be, for example, doing dry aging, but with some sort of maybe permeable bag or something of this manner that could also help. And then, as I mentioned before, crust formations can be sold and used to enhance the flavor of other food products and also create have the ability to create some high value products for consumers. Here are my references. I want to thank everyone who helped with this research and also all the funding and support that we received throughout this research. Thank you.
Thank you very much. All right, so again, to the floor for some questions first. Sure.
Yes, thank you. That was quite interesting. And I have two short questions. One is a bit more general. The context, I'm looking at the context of the consumer, let's say, acceptability of the product, looking at it from the angle that we are now getting more concerns on the global One Health thing, and we are having more and more cases of these zoonotic diseases, and this is game animals. So I was expecting that there would be some kind of concerns and also some rejection of such kind of work. I don't know how you have gone through that, if you have perceived that. The second question is more on the sensory tests you did. You used the same meat, let's say it's the same species, and you had an aspect of like beef, I don't know.
Beef flavor intensity.
Yeah, beef flavor intensity. I was wondering why you decided to use beef flavor intensity and not compare it with beef. Why couldn't you just say taste beef and you taste your different I don't know if it's flavors, and then you directly make a difference or a comparison to beef.
Okay,
Yeah.
Thank you so much for your questions. I will start quickly with the first one and say that the idea of doing commercial production with these animals is through having them farmed. So they're supposed to be farmed animals, yeah? Like specifically the ones that we use are actually farmed eland. So even if it's a game species, these animals, as I actually mentioned to the presentation, they're suitable for farming. So the idea is that the same way that we farm cattle, we would be able to also farm eland. If that answers your first question. Then, and in terms of zoonotic diseases, of course, if you have a typical farming system, they're going to be checked, right? And they're going to be taken care of and monitored. Like the animals that we have here in Lani, they are being monitored, even though they are a game species. And then for the second question, the reason why for beef, this is a very standard way of evaluating meat in general, especially for the game species. But the idea is also a form of familiarity. Most people don't know what game meat tastes like, but they have tried beef, for example. And in many cases, beef, it's not necessarily the same as eland, yeah, but it's still comparable in some ways, right? So this is why you have beefy flavor, which is considered something good, yeah? And like I mentioned, not most people have tried eland meat. And so that's why you have an overall gamey taste, right? And then you have your beef taste. It's similar to when you do rabbit. For example, rabbit, a lot of people say tastes like chicken, even though it's different, right? But the idea is to have something familiar. Any other questions?
Thank you for the presentation. I have a question from methods. How did you do the selection of 15 panelists?
Oh, that's great. So So actually the panelists were people from the Institute. And the idea is that they actually have tried already game species in general. So that's why they were trained, right? I mean, they're trained in that we gave them this, we told them how to do it, but also they have had more experience in tasting varieties of meat. So they were selected from the Institute to be able to do this.
Thank you.
Mm-hmm.
Glenys, thank you for your presentation. I have one very simple question. Maybe I missed that. What is finally the recommended time of aging?
Oh, that's great. So we did 14 days, which is what's considered kind of standard based on research. It has in the previous research, it was recommended maybe aging for 14 days would be better, but there's an upcoming study, which is focusing on perhaps elongating the aging to see what would be the best. So at the moment, 14, but it's still under investigation of what would be the right amount of time in terms for reaching tenderness goals, but also flavor development. Mhm. Thank you, guys.
All right. Yes. So we would like to just exchange the order of presentations due to technical details, and we would welcome first Sophie, and then we will move on to Ruth. And I hope she is available online and ready to go for her presentation next. Yes, I think the pointer is not working 100%. So if you do have some issues, you can use the keyboard.
Thank you.
Okay, so thank you.
So thank you so much. Let me shift your attention to matters of chicken in Kenya. So basically, this presentation is preliminary results for an ongoing project. So here I'll just be looking at assessment of natural antibodies and specific antibodies against Newcastle, but at a phenotypic level. So why this study? Disease is quite a challenge in most production systems in Kenya. And this is because this chicken, particularly in the scavenging systems, they're exposed to a variety of pathogens. Of importance is the Newcastle, which causes really high mortality rates, and there's production inefficiencies, and of course, matters of the economics, where there's a lot of losses, and also matters on low product quality. So we have biosecurity measures which have been put in place, but you often find that these measures are actually, the effects are actually temporary. So in this case, we're suggesting how about we do selective breeding for disease resistance or tolerance. But then again, disease resistance is actually quite a complex trait where you have aspects of host pathogen interactions and also environmental effects. So in this case, it's advised that you try and find indicator traits of disease resistance that are actually taking care of animal welfare issues and should be easy and cheap to measure. So in this case, we're considering natural antibodies where it's able to confer general health. And then we have specific antibodies where it can tell us whether these animals are able to respond to vaccination or immunization. So the objective for this particular study was just to look at what biological and environmental effects are influencing these antibody traits in our local chicken in Kenya. So to do this, we went out to the field, out in the farmers to get local chicken. And as you can see, we got what we call ecotypes. These are related to various geographic regions across the country. So, but based on microsatellite markers, we grouped these 13 ecotypes into 3 phylogenetic groups, as you can see from the map. So this was the first, actually what I should have mentioned, we used 2 populations. One is local chicken in Kenya, and then there's a synthetic breed that we tried to create within our breeding program. So within the indigenous chicken population that we got from the field, these are some of the genotypes that we were able to sample through because they are associated with various attributes. Then the second population, so here you can see we have the hybrids. We crossed them with our local chicken, and then we got the F1s. So within these F1s, we subjected them to intersame mating just to create some segregation. And then we realized that there was some two plumage colors that were quite dominant, and we decided to isolate and then breed amongst themselves. And then we ended up with the spotted and the black, and this is what I use as a synthetic breed. Then across both the local chicken and the synthetic, in this study, I used 5 generations, right from what we call the founding flock, what we got from the field, till generation 4, and these are subjected to selection to develop meat lines and egg lines. So these are already chicken under selection for production traits. So I'd like to also see if production is going to influence immunity. So just basically a quick one on the data that was collected. So assuming when an individual is exposed to an antigen, we expect the pathogen recognition receptors to recognize the antigen. Then thereafter you have the innate immune response being brought in, natural antibodies are produced, and the first one is the IgM isotype. Then through isotype class switching, we get the IgG and IgA, and on the right, you can see I've listed some of the functions of these isotypes. So once, in case the natural antibodies are not able to eliminate the antigen completely, there's an interaction between the two arms of immune where you have the adaptive immune response being brought in, and specific antibodies against that particular antigen are produced so that it's able to neutralize and eliminate the pathogen. So with that information, we sampled 1,540 chicken across the 5 generations. And this chicken ranged from 12 to 56 weeks of age. And then antibodies were measured using indirect ELISA. So to analyze, to test which factors influence these immune traits, a general linear model was fitted where the immune traits were the observations. Then you have a list of fixed variables. Then there's continuous covariate there as age, and then the plate effect as random. So just on to results here. We start with the natural antibodies. You can see that the highest, as expected, would be the IgM, because this is the first one to be produced, and also it's the major contributing isotype towards innate immunity. Then here you have the lowest being the IgA, which is quite important in mucosal health. So of course, in this case, you'd expect a higher proportion to be found on mucosal lining rather than in the bloodstream. Then the last one here, you can see that there's presence of specific antibodies binding to Newcastle disease, and this could indicate that the study population actually was responsive to their own station vaccination schedule that we have. Then just looking at which factors were actually significant here, the biological ones included sex, population, generation, and genotype. Then you look at the environmental bit, it was Hatch group and plate effect. Then age was significant as a linear covariate, which is an indication that immune function improved with age. And this is actually expected because as an individual ages, they're exposed to various environmental stresses, and this helps to shape the immune function. Then just looking at some of the factors that were significant here, you can see the effect of sex on immune could indicate that there's presence of sexual dimorphism among the antibody traits. But here you can see the females across all the isotypes actually were, the mean was higher than in the males. So this could indicate that the genes on sex chromosome have an effect, or also gonadal hormones does affect immunity. Then when you look at the two populations, considering that both of them were exposed to the same environmental conditions, variation in mean estimates could indicate that there's actually some genetic variability between the two populations, which is actually the case because one is indigenous to the area and the other one is synthetic, developed from various other breeds. And Also, the fact that the local chicken had a higher mean estimate than the synthetic could indicate that this chicken have a large plasticity in their immune system, particularly in the genomic regions that confer immunity. Then here we can see the genotypes I showed in the photo. The one that I call Kuchi is a game-gated genotype. That's when and the major genes that are associated with feather reducing genes actually had higher mean estimates for the two isotypes of natural antibodies, which previous studies have actually agree with this observation that we found. But then when we look at the Newcastle IgG isotype, the chicken with the normal feathered actually seemed to be more responsive than the other two. So in this case, we suspect that maybe there's some kind of negative correlation between the natural antibodies and the specific, such that if one has a higher mean, then if they have a higher mean for the natural antibodies, we'd expect them to have lower for the Newcastle. So maybe there's need to test this at a genetic level. So finally, here, there was an interaction between generation and line, and line here is the meat line and the egg lines that where selection was being carried out. So, but here we can see there's a general decline across generations, and perhaps it could be due to selection for production traits, given that production and immune traits have a negative association, and mostly could be because of the resource trade-off between the two functions. Then when we look at the interaction, there's a re-ranking of the lines across generations. So here, this re-ranking could show that probably the population is unstable, and this has something to do with the founding flock that we started this this research with, perhaps was not in linkage disequilibrium in the genomic areas where that affect immune traits. And in this case, this could mean that in the subsequent generation, the allele frequency was responding to any changes in the environment, irrespective of whether it was due to selection or production conditions. So in conclusion, we've seen that biological and environmental factors actually influence immune traits. And these factors need to be accounted for to reduce bias and improve accuracy when evaluating individuals, both at phenotypic and genetic level. And also the fact that there was variability among the population and the genotypes is something that we can actually exploit when we want to develop adapted and immune competent lines. So those are the references, and thank you for your attention.
Thank you very much. So I'll open the floor for questions. Just raise your hand if you have one. Okay. For me, it's not so easy to see the slides, so forgive me if I missed it, but I wanted to know about your founding group. Did you focus primarily on meat producing chickens or laying hens, or what was your
Okay, so
initial-
for the local chicken, they are they have not been selected for anything. These are just local birds where farmers do their own kind of selection based on just what they see in terms of the phenotypic performance. So what we did is, because there've been a lot of improvement programs through donor agencies that bring in hybrids to different parts of the country and encouraging farmers to actually crossbreed to improve their own local breeds. So what we did to try and avoid those regions, we tried to map out the areas where we know these development programs have gone to and tried to avoid them. So we went to extremely remote areas where the chickens are actually running outside during the day and then brought in during the night.
So as little selection as possible,
yeah,
basically.
so the ones that are there are probably due to natural selection, but not,
and utilized for, I guess, both meat and
dual
eggs.
purpose,
Yeah,
dual
okay.
purpose, yeah.
And then you mentioned later along your generations that the effect of selecting for production traits, in which direction did you go with that?
So what we could see when we tried to do the high because we're selecting for meat lines based on body weight, and then the egg lines based on age at first egg. So we isolate the high and the low for both sides, and then tried to see what direction is it going, and it was going the negative way, such that the ones with the high body weight, the immunity tended to go down. So that is why I was attributing this. Perhaps there's aspects of feed resources trade-off, because these are two functions that both have a metabolic cost.
And in your synthetic line?
Uh,
How did you?
Actually, it was in both cases, yes.
All right.
But the synthetic, remember, it's already was selected for. Yeah.
Yeah. And my last question is, you mentioned age as a factor.
Yeah.
For how long did you sample your animals? Like until what age approximately?
From 12 weeks to 56 weeks.
Okay.
Yes.
All right.
Mhm.
Thank you very much.
Okay.
If there's no other questions from the floor, thank you for a very interesting presentation.
Thank you.
And we will have to move to the online presentation, so technical assistance might be required. Okay, so first we'll double check if she's there. Okay. And it will be, our last presentation to do with monogastrics. There we go. So Ruth, can you hear us, and can you activate your microphone so we can hear you?
Yes, I've done that. Can you hear me? Okay, you can hear me.
And you can also try to upload and share your presentation with us, please. All right, well, I could see she was active because I think she unmuted herself, but she tried to promote. Ah, there we go. I think I heard something. You can just turn it up. Yeah, so in the meantime, if you can load your presentation, that would be good.
I'm trying to do that. Okay, I clicked on the wrong one. So I've gotten it now.
Okay, we're just seeing if we can promote you to panelist, then you should be able to control.
Okay, because I need to share my slide and it's not showing on my screen, my Zoom screen.
Oh, it's not visible.
It's not showing on my Zoom screen. My Zoom screen
Okay, we're going to try a different approach to see if we can invite you.
is the place to share slide. Okay.
We're still working on it. Sorry for the delay. Oh, so the wrong one. I think you added. It should be this one at the bottom. Oh, okay. Okay. Okay, so it looks like you've been promoted to panelist, which is good. And if you can try again to load your presentation, please. Yeah, from our side, it should be working.
Okay. So now I can share my slide. I'm coming. Let me open it.
Okay. Yeah, Ruth, we also cannot hear you, so please talk louder or come closer to your PC microphone.
Okay.
Yes.
Can you hear me now?
Yes, we can hear you now. And if you can look at your screen, we're indicating where you can share it. It's the big green button.
Yes, I've seen the green big button.
Okay.
So I think I'll just I'll open the slide properly since I've already prepared it. Just give me a few seconds.
Okay, in the meantime, while you're figuring that out, Ruth, I would also like to remind everyone that we have a poster session linked to this theme, and it's starting at 6:45 in the round hall. So we will be using the usual format and going from poster to poster, so you can have some opportunity to ask the presenters some questions there. Okay. Ruth, can you try also email the presentation to us if you're not able to load it? Marek will tell you the email address.
Okay.
Or he will write it in the chat so you can copy it directly.
Okay. Okay.
And the other option is to attach the file in the message directly.
I've seen it.
And then I will change the slides if she's sure. Just double check. Yeah. If someone's coming through. All right, Ruth, we're gonna have to give you just a couple minutes to sort it out. Yeah. Otherwise we're running really out of time. Also, another reminder for everyone that this evening is the conference dinner with live music. So if you booked your tickets, it will be in the refectory, or what we call Mensa canteen system. So welcome to join us there. And afterwards, there will be an after party in Club C, and this is free for everybody. So you didn't have to book that previously, and welcome to join us for some more live music and socializing and networking. Ruth, have you sent us anything yet? We'll have
Yeah,
to make.
I'm trying to send it. It's I think that has to do with the network if it's strong enough.
Mhm, mhm.
I had pre-recorded something, so I think I need to shut down some things here. Just give me a minute. A few minutes. Okay. So this is the original one. Okay.
Otherwise, Ruth, I think last case scenario is if you would lead us through your presentation nonetheless, we can listen to you and we can still ask you questions if that's
Okay.
possible. All right.
I have a okay. Maybe I'll just do that.
Okay, so just remember to speak clearly and loudly into your microphone so that we can all hear you. And you can start when you're ready. All right.
Okay. I might just use the PDF I prepared. This is not a PDF.
So you can go ahead. We're ready for you.
Okay. Can you see my screen?
Yes, I see. It says has started to share screen. So perfect. We can see your presentation.
Okay. So
So you can start.
can you hear what's
Yes. Yeah, we can see your slides moving and we can hear you.
oh
Okay, so will you go ahead and start your presentation, please?
can you hear? Or
Yes, we can hear you, but yeah, you can go ahead and start your presentation.
okay, because I've seen what is there with you. Then the introduction.
Yeah, perfect. Now we cannot hear you at all.
Oh, that's serious. Can you hear?
Now, yes, but we cannot hear your presentation, so we can just hear you asking if you can hear us.
This is what I'm going to do. I'll share the PDF version of it and then go through it. I think that's the best way forward.
All right, we only have a couple of minutes left, so what you can do in that time possible would be great.
Okay. Can you see the screen now?
Yes.
My slide is showing.
Yes.
Okay. So my name is Ruth and I'm making this talk. I'm making this talk on the effect of Ocimum gratissimum on egg quality of laying hens. This objective of carrying out this study is to determine the effect of Ocimum gratissimum on egg quality when it is consumed as an aqueous extract or as a component of feed for laying hens. This is a brief outline of the presentation. I'll introduce the topic, then discuss the materials and methods, the results, give a brief summary, and conclude on the study we carried out. Eggs are a source of good quality protein for human consumption. It is considered as a complete protein because it contains all essential amino acids required for human growth and development. Egg qualities such as shell thickness and yolk color are of benefit and importance to both farmers and consumers of eggs. Reason being that to a farmer, cracked eggs and eggs with broken shells is an economic loss. And for consumers of eggs, when an egg to be purchased is cracked or is broken, that is not economically wise to purchase. Most consumers will not want to purchase such eggs. Then in addition to this is the color of the yolk. For consumers of eggs, not all consumers are concerned about the color of the yolk, but some consumers emphasize on the color in that they would love to buy eggs that have bright yellow colored yolks. Now, plant extracts have been reported in literature to elicit positive effect on egg quality. And the plant we use in this study is Ocimum gratissimum, also referred to as scent leaf locally here in Nigeria. It is a perennial medicinal plant with biological and physiological properties useful to both humans and animals. So how did we go about preparing this plant to incorporate it into the feed of laying hens? What we did was that we obtained a large quantity of the plant of Ocimum gratissimum here, the fresh leaves. Then we rinsed it in clean water, allowed the water to drain, then separated the leaves from the stems. After doing that, the leaves were chopped into small particle size. Then 1,000 grams of the leaves was weighed with a digital scale, milled with 750 mils of clean drinkable water. In this picture here, the already milled leaf is being sieved with a cheesecloth to obtain the liquid fraction, which we use as the aqueous extract. Then the civet in this container here, we refer to it in this study as the shark. It is sun-dried here, then packaged into Ziploc bags before it is grounded into fine particles. Then these fine particles are incorporated into layers match, which is the feed for the birds. And it is included at an inclusion rate of 50 grams per kilogram of feed, while the aqueous extract here is withdrawn into 1 ml syringes for administration to the birds that were placed on the aqueous Ocimum gratissimum extract treatment. Feeding trial and determination of egg quality. A total of 60 bovine brown layers at 22 weeks of age were randomly distributed into 3 treatment groups, having 5 replicates and 4 birds per replicate. Feed and clean water was provided for the birds. The eggs were collected on a daily basis for a period of 8 weeks. The experiment was terminated on day 56. And on day 56, 3 eggs per replicate of total eggs collected on the day 56 were used to determine egg quality according to the method of Abdul Wared and Lohakari 2020. Data collected on the egg quality was subjected to a one-way analysis of variance, and significant means were separated using Duncan's multiple range test. Proximate analysis was carried out on the composition of the experimental diet. And from the results we obtained in table 1 here, the crude protein content of the layers mash we used for the birds was adequate in that we got 15.76 grams per kilogram dry matter, and the requirement for layers is 16 grams per kilogram dry matter, while the crude protein content of Ocimum gratissimum shaft was 12.29 grams per kilogram dry matter, and its nitrogen-free extract concentration was 61.60 grams per kilogram dry matter. The results on egg quality are presented in table 2A and 2B. Although there was no significant difference across the treatment groups with regards to egg width, shell thickness, albumin weight, and albumin height, it is proper to emphasize or is noteworthy to mention that the albumin weight and albumin height was numerically higher in birds that consumed Ocimum gratisinum, either as the aqueous extract or as the shaft, which was made part of the feed, that is as a component of the feed. This same pattern was observed in the yolk quality. Like here, the yolk weight and the yolk width also was numerically higher than that which we recorded for the control group. Then administering 1 meal of aqueous Ocimum gratisinum to laying hens resulted in better yolk color compared to the control group and to the birds that were fed diets that contained Ocimum gratisinum shaft. In summary, consumption of Ocimum gratisinum by laying hens did not significantly increase weight. Numerical differences were observed in albumin weight, albumin height, yolk weight, and yolk width in eggs from laying hens that consumed Ocimum gratisinum, either in feed or as an aqueous extract. Aqueous Ocimum gratisinum improved yolk color in laying hens. In conclusion, it can be said that administering 1 meal per bird of aqueous Ocimum gratisinum extract improved yolk color in eggs from laying hens. These are my references. I would like to acknowledge the students who participated in this project for the work done and the time taken to prepare the leaves before it was administered to the bird. Thank you that come from me. Thank you. So I'm done.
Thank you very much, Ruth, and thank you
I was
for
just running through it.
thank you for persevering through the technical difficulties.
No problem. Thank God.
I would like to open the questions to the floor, if there's anything. Okay, there's a question.
Hello. Thank you, Ruth, for your presentation. I was wondering how did you pick the 0.1 ml dose, like based on some studies, or how did you select the dose? And the second question is how did you feed the chicken? Because it's a liquid, how like you feeding it through the oral, or how did you do?
Okay. What we did for the aqueous extract, we had carried out previous experiments with the same plant, Ocimum gratisimum. And in that experiment, we prepared the aqueous extract, put it inside the drinking trough, and allowed the bird to consume as they willingly on voluntary consumption for a period of about 3 hours. Thereafter, we withdrew the aqueous extract from the birds and give them normal water. And that was a broiler experiment. So what we observed was that some of the birds did not quickly go and start consuming the liquid. They stayed for some time. Then after they started consuming, maybe because of the black color of the liquid. So we now determined that, okay, rather than allowing voluntary consumption, which we may not be able to say this is the exact quantity that each bird in a particular replicate group to consume from the liquid, we decided to bring it down to 1 meal per bird, because we know from literature that Ocimum gratisinum, the aqueous extract, it has the capacity of lowering blood glucose when it is consumed at a high amount. So we just limited that small, with that information at the back of our mind, we decided to do 1 mil. In the previous studies we've done with broilers. Then when it came to this layer study, we decided to just stick with that 1 mil rather than increase.
Thank you.
Okay, so we've used the full time of this session. So I'll have to wrap it up. Thank you very much, Ruth, for persevering and presenting to us. And thank you very much for attending the session, and enjoy the rest of the conference. Hopefully see you at the poster session.